I'm Helen Evans. I'm part of an artistic duo with Heiko Hansen. We've been working for over 20 years now, collaborating on artistic performative installations, often on environmental issues. We've worked a lot on the issue of air pollution, where we use we represent air pollution through the metaphor of the cloud. And in our projects, we often collaborate with other people, whether scientists or researchers or NGOs. Working with INL, which is the International Nanotechnology Institute based in Braga, in Portugal, is a new departure for us, and it's part of a project that is called O.R.S, which is a project where we're trying to address the issues of water pollution. So the O.R.S is an abbreviation for the Orbital River Station, which is inspired by the von Braun rotational space station wheel, except that it's a large floating form which is based in a river and changes with the water currents. So this is a rendering of one of the versions of the O.R.S. Its form and its colour resembles a giant life ring, which is used to rescue people in the water. And the project is being developed as part of Big Torrent, which is a programme conceived and produced by Bipolar, which is a project which invites artists to respond to the issue of flooding, of sudden water rising, and risk prevention along the River Rhone. And so the O.R.S, we're currently about to enter the production phase of this object, and it will be shown in the summer in Avignon, Arles, Lyon and Chalon. And it's a kind of environmental machine, a kind of laboratory for observing and monitoring and capturing contaminants in the water. It's also a multifaceted art project. So the idea about the O.R.S is that this is a 3D model that we made of it. Here's another model which the idea of this O.R.S is that it should actually rotate in the water, and the rotation is created by some pal, like a form on the bottom of the ring. Sorry, I'm forgetting my English now. Which enables the current of the river to turn the ring. It's a small-scale installation, so it's 5 meters diameter. And we're hoping that this rotation, theoretically, one day could be used to actually generate energy, which would then power the water monitoring devices on board. So the water monitoring devices on board are a mixture of analog and digital. And so when we first came to the Nano to Water group, which is the laboratory who's hosting us at INL, we were very excited to discover the way that they use nanotechnology to monitor and capture specific water contaminants. But they also investigate the potential toxicity of nanoparticles themselves. So they're operating on two different ways, looking at the positive aspects that nanoscience can bring, but also the potential dangers. And that was a very unexpected and interesting discovery for us. And so we have developed 3 kind of axes of research with them. This is a photograph from a test we made with our models in the fluid mechanics laboratory at the University of Le Havre, where this is a simulation of a very large, long aquarium, about 12 meters long, where you can simulate a current. And so we could verify that the model turns. Of course, the model turns, but whether the 5 meter turns is another matter. But we shall find out. That's why it's a research project. So in the Nano to Water group, they have developed in research phase a paper, so now I shall go on to this slide, a paper-based testing kit for on-site water quality monitoring. And what they use is they synthesize nano-gold particles, which they cap with in a particular chemical process. The solution is functionalized to detect a contaminant. So they can functionalize the nano-gold dots to react to the presence of sulfide or the presence of lead. And then afterwards, what they do is they print in wax onto a chromographic paper, leaving small dots onto which they place their nano-gold solution. And afterwards, you can then apply your river water sample onto the dot, and if you shine UV light onto your dot, it will either fluoresce if your water is clean, or it will stop fluorescing if there's a presence of a contaminant. And they can then measure the quality, the quantity of that contaminant. So normally, this research project, these dots are usually arranged in a grid. There's no formal composition which is considered in the scientific lab. So we propose to start to arrange the dots across the paper to make a map of the river. So at the moment we're experimenting with, we've mixed up some of the chemical solution here. You can see that the bottle on the left has got some contaminated lead in it, and the bottle on the right hasn't, so it's glowing. And then with these dots, we are experimenting at the moment with placing them onto various maps. We're experimenting with trying out different kinds of papers and different kinds of techniques to protect the paper from the nano-gold, to control where it goes in order to make the drawing. We'll be using the O.R.S. boat vehicle this summer to take samples from the river on the Rhône. For the moment, we work with the Valley of the Seine. So this is a map of going from Le Havre to Rouen. The river is represented by its geography, and it's rendered visible through its water quality. For the moment, these the nano-gold that they've developed in the synthesis of the chemical solution in the laboratory can detect only sulfide and lead, but we'll be also looking at other ways of capping the material to capture other contaminants. We have begun collecting water samples along the Seine Valley, and we ran a workshop with students from the Art, Media, Environment program, which we run in Ezada, which is the art school in Le Havre. And the workshop was part of this research project to look at the interaction between man-made environments and wildlife. And this workshop took place in Harfleur, which is a very picturesque town, which is situated very, very close to the centre of the industrial heart of the industrial zone of the Le Havre metropole, where there's petrochemical and pharmaceutical factories. So despite the presence of very heavy industry, we can see that nature adapts and resists in very surprising forms. And here we could call it an automatic bio-human sculpture. So besides using nanoparticles to detect contamination, Nano2Water group, which is a group of about 10 mostly chemists and marine biologists, they're also conducting experiments to test or to establish under what conditions nanoparticles could be toxic when they find their way into the environment. And indeed, silver and titanium oxide nanoparticles are increasingly found in our rivers because they're used in consumer textiles, paints, cosmetics, and our food. So in the laboratory, what they do is they run experiments on zebrafish. Zebrafish are then bred and they collect the eggs. And then using the embryos, they expose the embryos for a period of 72 hours, because that's the legal limit at which you can test on a zebrafish egg. Expose them to various degrees of different sizes of nanoparticles, of different. There's the problem with nanoparticle, the possibility of toxicity in nanoparticle science is that there's so many different kinds, and they all change depending on their size, the kinds of enzymes and proteins that surround them, whether they coagulate or not. So there's an infinite range of possibilities. So they do very elaborate, lengthy experiments with the zebrafish to see whether they grow at the right rate, whether they have cardiac problems, whether they live, or whether they're deformed. But of course, these experiments are done on kind of levels of titanium oxide in various quantities, which don't necessarily have a cultural relevance to people who might live next to a river output, a river sewage output. So we would like to also do some experiments with them in collaboration, where we take water samples from particularly polluted zones, which we imagine would be very polluted, and expose the zebrafish eggs to that polluted water to see, is it possible, seeing what the effect would that be on the zebrafish, to then conclude what may be in the water. And the third final project we're doing is thinking about a way of appropriating and repurposing the scientific methods. So here it's really about image making. And the technique, one of the things we found very fascinating in ENL is the techniques of microfluids that they work with, which they use to develop electrochemical biosensors, which are highly complex systems for. And we were wondering whether this could be used as a process of generating an image which works on a more poetic level, which perhaps can encourage empathy. So in our research, into the water quality in the Rhône Valley, we learnt about a little-known species which is called l'apron du Rhône, the apron fish. He's a very shy fish. He's not particularly beautiful. He's not particularly delicious to eat either. But the apron fish used to be found everywhere along the Rhône Valley, along a stretch of 2,000 kilometres. Today, it's an endangered species, and there's only 200 kilometres left of habitat for this fish. The fall in numbers is actually symptomatic of the destruction of its natural breeding ground, and of course the quality and the quantity of water available for him. So he's a kind of bioindicator, if you like, for water quality. The other interesting characteristic about the apronfish is that he has an ability to change his colour, to camouflage himself with the riverbed, and his defence mechanism is to become immobile. So we're, at the moment, prototyping a kind of installation performance image, we call it, which depicts an apronfish using the technique of microfluidics. So here you can see a microfluidic form where you can pump through a blue ink, and then you can follow on with a yellow ink, and you can change the colours, so you can have the ability to, if you layer several of these together, to create an image. So we're experimenting with this. So just like the real apronfish, the image has the ability to change colour, to appear and to disappear, depending on the flow of the fluids seen through a series of laboratory pumps. And it's a kind of metaphor for our relationship to other species. And that's it. Thank you.